Hi folks, today we're going to talk about heredity and Punnett squares. Well, first off, you might want to know what genetics is. Genetics is just the study of genes. So there's a long history that goes with all of this. We'll start with around 1856 to 1863. We see that Gregor Mendel discovered the idea of inheritance, or the idea that parents pass on genes to their offspring. In 1882, Walter Fleming discovered chromosomes when looking at cell division. At the time, he didn't know that chromosomes were responsible for passing on genes. In 1908, Thomas Hunt Morgan was the first to realize that chromosomes pass on traits. And then in 1990, we have the Human Genome Project, which set out to determine what exactly each of our 20 to 25,000 traits do. And they did finally complete that in 2003. Some quick vocabulary for you. We have heredity, which is the passing on of genetic material, or genes, from parent to offspring. We have characteristic, which is a feature that has different forms in a population. For example, eye color, hair color, freckles, etc. These are associated with genes. Traits are just the different forms those characteristics can take. So for eye color, you could have blue eyes or green eyes, or hair color, maybe you have brown hair or blonde, etc., etc. These are associated with alleles. Genotype, that's actually your genes, or the allele pair that you have for a characteristic. Phenotype is how that allele pair presents itself in your body, so the observable trait that you have due to those genes. And probability, which we'll get to towards the end, that just expresses a ratio or a percent that's a predicted chance of some kind of outcome. So it doesn't mean that this is exactly how it will happen, but it's the predicted possibility of something happening. All right, so let's look into this by thinking about the experiment that Gregor Mendel did. He was a monk in the 1800s. He lived on a monastery, and he studied the pea plants in that monastery's garden. And he measured all sorts of different characteristics of the pea plants. For example, he would measure how tall the plants got, the position of the flower, was it on top of the flower or coming off to the side? The shape of the seed, was it a round seed or a dimply seed? The seed color, whether it was green or yellow, the shape of the pod, the color of the pod, and the color of the flower. So obviously Mendel had a lot of times on his hands. He was measuring all these different characteristics of all these different plants. And furthermore, he even started breeding them together to see what kind of traits might be passed on. So he took a green plant that was true breeding, which means that if it was self-pollinated or it reproduced on its own, only green pea plants came out. And then he took a yellow true breeding pea plant and he bred the green and the yellow one together. And what did he get as offspring? What might you expect? You might expect that he'd get some green and some yellow, about half and half, because you have a green mom and a yellow dad. But as it turns out, every single one of the offspring from the green and yellow plant were all green, no matter what. He tried a lot of things, and he couldn't get a yellow plant to come out from the reproduction of those two true breeding plants. So he did some further experimentation. So he took that first generation of pea plants, all the green ones that he just made, and self-pollinated them, which means that he reproduced them on their own. And he noticed that when he took up all of the new offspring and counted up how many were green and how many were yellow, he had about 75% of them were green and about 25% were yellow. And that was weird because he just took a green and yellow one and only got green out. That yellow trait had seemed to fade away. It was gone in that generation of plants, but now it came back. So from that experiment, Mendel decided that green was a dominant trait because it was the one that happened most often and it was the one that took over completely in that first generation. And then he decided that yellow must be a recessive trait because it receded or it went away in that first generation. He also decided that from each parent, there must be some type of factor that gets passed on from the parent to the offspring. So at the time, he didn't call them traits, he called them factors, but he theorized that offspring get one factor or one trait from each parent. Now, we know that we do get those factors from our parents, and we call them genes. Quick recap before we move on. Heredity is the passing on of traits from parent to offspring. Both parents have two alleles, and they give one of those alleles away to their offspring each time that they reproduce. That offspring receives two alleles because it gets one from each parent. So now that we know that, we can think about genes and alleles and predicting the probability of certain traits being passed on. We represent genes with letters. And it can be any letter, it doesn't really matter. We just kind of pick a letter. And if it happens to be a dominant trait or a dominant allele for that gene, we use a capital letter. And then the recessive side of that gene, we use it the same letter but lowercase. So if our pair of chromosomes within the genes that we have the same type from both of our parents. So we have one dominant from our dad and one dominant from our mom. We call that a homozygous allele pair. Or it could be that they're both recessive, and that is a homozygous recessive allele pair. If the gene has two different alleles, for example, one dominant and one recessive, those come together and make a heterozygous allele pair. When you have a homozygous dominant or homozygous recessive allele pair, it's very easy to determine what phenotype you're going to have. It's obviously just going to be whatever allele pair you have. So if it's dominant, you're going to have the dominant phenotype. If it's recessive, you'll have the recessive phenotype. But if you have a heterozygous allele pair, sometimes it can be tricky. Most of the time, and especially when we're just learning about it, we think of complete dominance, which means that if you have a dominant and a recessive allele pair together, or a heterozygous allele pair, the dominant trait will take over and the recessive trait just gets carried. It doesn't actually show up in the phenotype. However, there are two other types of dominance that can happen. Incomplete dominance gives you a blend of the phenotypes. So for example, if your flower had an allele pair that one allele said it should be red and the other said it should be white, you're actually going to end up with a pink flower, so it blends the two together. There's also codominance, where both of them show up equally. So in this instance, you'd have red and white would make maybe red and white stripes, or white with red spots, or something along those lines. It wouldn't show up pink, and it wouldn't show up just one color. Both colors would show up. We do have the ability to predict what kind of inheritance will occur. So depending on which alleles the parents have and which alleles they give to the offspring, that offspring can have a few different types of genotypes. To predict those different genotypes, we use a Punnet Square, which is kind of like a multiplication table, but for traits. The Punnet Square was developed by Reginald C. Punnet, who literally wrote the first book on genetics. It does not tell you with 100% certainty what offspring you're going to get, but it does give you a probability of what traits will be passed on to your offspring. So for example, if it says there's a 25% chance of your child having cystic fibrosis, that doesn't mean that 1 in 4 of your children will have it. It means that each time you have a kid, there's a 25% chance that they will. When we talk about probability, we could use a ratio or we could use a percent. And we usually use a percent when we look at this. In this case, each of our 4 boxes in the Punnet Square represent 25%. So if we take those pea plants, for example, we could look at that one gene that codes for color. And remember that we could either have green or yellow pea plants. So remember, we get 2 alleles, one from each parent. So the original true breeding plants, the green ones, would have 2 letters that go with their genotype, or what genes they have, what allele pair they have. In this case, since they were green, we decided that we're going to use the letter G for this allele. And since green is the dominant trait, according to Mendel, we'll use a capital G. And then for the yellow one, since it's recessive, we'll use the same letter but lowercase, so 2 lowercase g's. And they're the same, so we have 2 homozygous parents, a homozygous dominant and a homozygous recessive. Mendel bred these 2 together. And if you think about it, let's think about this the math way. If you have, let's see, dad is capital G plus capital G times mom, which was lowercase g plus lowercase g. If we use the FOIL method, we take the first times the outsides times the insides times the last. No matter what we get, we're going to get a big G little g both times, or all 4 times. We have big G little g, big G little g, big G little g, big G little g. So that first generation of offspring are all big G little g, or heterozygous. They have a heterozygous allele pair. Since big G represents green, and green is the dominant trait, they all showed up as green. That recessive trait just gets carried on. It doesn't actually show up in the phenotype. Now, if we self-pollinated one of these plants, or we made it reproduce with itself, we can do the same thing. And when we do that, we get big G, big G, big G, little g, big G, but we always write the big one first anyway, so it's just the same as the last one, and little g, little g. So now we have 1, 2, 3 different types of offspring we could end up with. This one will show up as green, since it's both green. This one will show up as yellow, since they're both yellow. And this one shows up as green, since green is dominant over the recessive one. So now, out of these 4 plants that we made, 3 of them were green and only 1 was yellow. So the probability of ending up with a green plant is a 75% chance. Finally, for our last one, we'll cross 2 flowers together. The first one is heterozygous for blue, blue being the dominant trait here. In order to be yellow, it has to have 2 recessive, so it's homozygous recessive. So we put the blue flower up here, and the yellow flower down here. And when we cross them, we get this. Notice that now we have half of them are blue, half are yellow, so there's a 50-50 chance of being blue or yellow. And that is in the example of complete dominance. So for complete dominance, there's 50% chance of blue, 50% chance of yellow. If it were incomplete dominance, there'd be a 50% chance of yellow still, because these are definitely yellow, but blue and yellow mix together to make green. So for incomplete, since they're blending together, it'd be 50% of that is a chance of being green. And if it were codominant, again, still 50% yellow, because homozygous doesn't play into the codominance or incomplete dominance. But these 2 would show up as blue and yellow, like maybe stripes or spots. So you have a 50% chance of being blue and yellow. And I'll leave you off with a pun. I certainly hope that you've learned a little bit today.